Our next speaker is Dr. Monique Smith. Dr. Smith did her postdoc work with Dr. Robert Malenka at Stanford University. Now she is an assistant professor of neurobiology and neuroscience at the University of California, San Diego. Dr. Smith's lab applies modern circular neuroscience tools and behavior tests to study pain, empathy, and neuropsychopharmacology. And today she will talk about protocols for the social transfer of pain and analgesia in mice. So Dr. Smith, the floor is yours.
Thank you so much for that kind introduction. Let me get my PowerPoint pulled up here. All right, thank you everybody for coming today. I'll be talking about our work developing the rapid social transfer of pain and analgesia in mice. And as was mentioned, I was at the University of San Diego. Now I'm at the University of California, San Diego. Oh, let's see, sorry, technical difficulties. So in our lab, we're really interested in understanding how the brain implements social behaviors and how do different social behaviors influence the neurobiology of the brain. And we look at this in all sorts of different ways. Today, with the focus of the protocol, we're going to be really focusing on how we explore empathy. And this also relates to ways to look at social communication of affective changes. All right. Oops, sorry, a little bit of a issue there. So for the talk today, I'll be giving you a little bit of background to tell you about the purpose of this study and the purpose of developing this protocol, and then walk you through the step-by-step protocol for the social transfer of pain and analgesia. And I welcome any questions about this, about how to implement this, or how to modify it for different types of tasks. Okay, so empathy, I think, can be defined with a cross-species definition so that we can actually see empathy in rodent models. And so if you think about it like the adoption of a sensory or affective state that is more appropriate to another situation than to one's own, then it's very clear that this type of empathy is seen in all different sorts of animal models. Sorry, I keep having issues with my screen. So we know that empathy is a core social ability for humans, and it plays an essential role in the social communication of pain and all different sorts of emotional states and sensory states. And it involves different integrated behavioral, cognitive, and emotional processes. And if you think about empathy from the nested doll sort of model, we have basic models of empathy like motor mimicry and emotional contagion that are sort of evolutionary conserved in many mammals. And then though the sort of higher levels of empathy depend upon those, like empathetic concern and consolation. And then on the very outside, when we get up to primates and humans, we have things like perspective taking and targeted helping. At least that was the idea in the literature. Until about a decade ago, people started wondering, can we really see different forms of empathy in the rodent beyond emotional contagion? So I would argue that seeing empathy in the rodent isn't actually that novel. If you look back into the literature, there is evidence of empathy all the way back to the 1950s, where it was shown that rats will forgo pressing a lever for a reward if it's contingent upon shocking their cage mate. And nobody called this empathy at the time, but I would argue that this really does have to do with empathic processing in the brain. In addition, although people weren't really calling it empathy, we've seen everything from behavior matching, social analgesia, social transfer of sickness behavior, and even helping and consolation behavior in rodents. And so if you look at it this way, we see all of these levels represented in that Russian doll model of empathy, from emotional contagion and mimicry to consolation and even helping behavior in rats. So one of the first studies to really say that we should start using mice, in fact, as a model of empathy, was from Jeff Mogul's lab by Langford and others. And in this study, they showed that mice injected with acetic acid and are isolated, they show very stereotypical writhing behavior that we're familiar with. And if you pair them with a mouse that did not receive any injection, this will actually dampen their pain phenotype. But if you pair them with a mouse that also received acetic acid injection, this will enhance their pain. So they called this the social modulation of pain and argued that we can use this to study the neurobiology of empathy. And then probably most definitely the most common form of empathy in the rodent that's used to study the neurobiology of empathy is observational fear, or the social transfer of fear paradigm. So some of this work was first published in the Lavis lab and by Hee-Sup Shin back about a decade or so ago. And in these paradigms, they show that mice observing either a familiar or unfamiliar mouse get shocked will demonstrate fear behavior. So once this mouse is shocked, this mouse learns solely from observing the other mouse to be afraid. And so this really is, when you think about that definition of empathy, this is, the mice demonstrating behavior that's more indicative of another situation than their own experience. And then that leads me to some of my own work during my PhD, where we sort of serendipitously stumbled upon something we ended up calling the social transfer of pain. And what we were doing in these studies is looking at alcohol withdrawal and measuring pain during alcohol withdrawal. And all the mice were individually housed to allow for alcohol drinking with wire cage tops and no filter tops, no ventilated racks in the room. And what we found was that if we took the alcohol away for 24 hours, the alcohol mice really showed robust hyperalgesia. But the interesting thing was that all of the water drinking mice housed in the same room, but individually in their own cages, also demonstrated hyperalgesia. And we spent a lot of time characterizing this to try and determine, was it the time of day? Was it the housing situation? What could this possibly be? And we finally determined that this really was the social communication of pain and the existence of olfactory cues in the room that was leading to pain in the control mice. And so this was sort of the development of the social transfer of pain. And we did this over the course of a lot of studies. We showed it with different types of drug withdrawal, so alcohol withdrawal, morphine withdrawal, during chronic inflammatory pain. We showed it in mice and also in prairie voles. And so we did a lot to really characterize this and look at the social communication of pain in these studies. But there were a few problems with this paradigm. When we went to study the neurobiology using chemogenetics, we realized that you have to have these mice all individually housed, which, takes up a lot of cages, takes up a lot of space. In addition, you had to have the control mice housed in a completely separate, but as identical as possible housing room, meaning at certain times I had 4 or 5 different behavioral rooms dedicated to just me, which is not possible for most researchers. And there was a long time course. So the mice were in withdrawal for a full 24 hours. And that looking at that 24-hour time course isn't really ideal for using things like optogenetics, which, we really need to have a short stimulation parameter paradigm. And so with that, in the Monique lab, we wanted to really modify it and further look at the neurobiology of the social transfer of pain to convince people that this could be a good model of empathy and really look at the neurobiological mechanisms. And so what we wanted to do was to design a paradigm that was easier, quicker, and more accessible to other researchers. So it didn't require multiple separate behavioral rooms. It was on a faster time course so that we can combine this paradigm with advanced optical neurotechnologies like calcium imaging, optogenetics, etc. So that leads me to the protocol that we published in STAR Protocols recently for the social transfer of pain and analgesia. So this is just the basic timeline that we display in the paper. And this was based off a paper that we published earlier in 2021 in Science. And so first, you just need to acclimate and handle the mice. You need to be familiar with different testing paradigms that you want to do. And it's a relatively straightforward procedure, I'll say. And so the second step is that you assign them to the different groups, which I'll get to in a moment. You only have a 1 hour social transfer of pain, and then you do all your behavioral testing tasks that you're interested in. So before you begin, you need to have the experimental setup, which I mentioned is like relatively straightforward. And I'll go through in a moment. And you need to have your behavioral testing apparatus and your mice all ready to go. And so we use sex-matched litter mates. We haven't tried pairing mice together of different sexes. We always do males with males, females with females. They're housed 4 per cage, and there's a specific reason with that I'll get to in a moment. They're given food and water just freely. They have free access, so there's no need to food or water restrict. Our mice are on a 12-hour light dark cycle, but we have done these experiments both during the light cycle or during the dark cycle, and they work just as well. And this is really like a critical component, is that experimenters really need to be extensively trained in mouse behavior. Anyone that's a behaviorist knows that the mice can really be easily stressed, and different ways that you handle them, especially if you're combining this with optogenetics and tethering to them to wires and things like that. You have to be really sort of gentle with them and move them in ways where they're not going to be too stressed out. And the main assay that we use is the mechanical sensitivity assay, which I'll also talk about in a moment. But experimenters need to be really familiar with this and have practiced quite a lot before conducting these experiments. And so for the mechanical sensitivity paradigm, the Von Frey hair stimulation, you can either create a rack yourself, or you can buy these from places like BioSeb, which we've listed in the protocol. But this rack that we actually created is just from Home Depot, so like a home improvement store that we modified ourselves. So it's relatively inexpensive and accessible for anyone who wants to build it. And then we have clear plexiglass boxes in which we place the mice on top of a wire mesh. And so this is so that you can have access to the bottom of their paws, which you'll stimulate with the Von Frey hairs. And so these boxes are relatively small. They fit 1 mouse each, and you're able, and these mice can see each other, hear each other, smell each other during this process. And so that is one critical component that I'd like to mention, are the olfactory cues. And so you really need to keep your control mice away from your experimental mice, from any mice that are receiving any pain injections, pain stimulation, even the mice that acquire pain. And that's because we found through a lot of extensive testing, some of which is published, some of which is unpublished, that cages, bedding, and really any olfactory cues from experimental mice that have experienced pain can lead to pain in control mice. And that was really how we accidentally found that original phenotype. And so if you can see here in this panel, we showed in the STAR protocols paper that if you even test mice on the same rack as mice injected with something called CFA, which is an inflammatory medium, or with the bystanders that have acquired pain, this leads to a marked and significant hypersensitivity compared to their baseline. In addition, we've done some recent studies, which are unpublished, that show that if you take bedding from mice that are just control mice that have no pain induction, this doesn't have any effect on other mice. If you expose mice to this bedding for 1 hour, they look relatively normal. So their mechanical threshold is represented here on the dotted line at baseline. And you can see that 1 hour exposure to the olfactory cues from other mice has no effect. But if you expose naive mice to bedding from mice that have been injected with CFA, this caused, just for 1 single hour exposure to bedding only, there are no other mice in the room, this leads to a marked hypersensitivity. So you can see this decrease in the mechanical threshold is actually representative of hyperalgesia or enhanced pain sensitivity. So you have to be very careful, make sure you have very good air filtration. Trash cans and everything need to be outside of the room. You need to clean really well and make sure there are no olfactory cues for the mice to communicate this pain to each other. So with that, I'll get into the actual step-by-step procedure for the social transfer of pain. And so 2 days prior to assigning them to groups or giving them any stimulation, we do our handling procedures. And this is just to bring them into the room, allow them to habituate to the room. We allow them to sit in the room for about 40 minutes or so. And then any behavioral testing apparatus that you're going to put them on, so I'll use the Von Frey as an example, we'll put them up on the Von Frey rack, allow them to sit there for 20 to 40 minutes or so, and get exposure to that, as well as get exposure to the actual experimenters that are running the experiment. It's very important that the experimenters that are handling the mice during these procedures during acclimation are the same that are doing the testing during the testing phase. So after these 2 days of habituations, we do a baseline Von Frey testing to see what the mice look like in a normal state. So what is their baseline Von Frey mechanical sensitivity? And then we assign them to different groups. And so this is why it's really important that we have mice housed mostly in even numbers, so either 2s or 4s. And so here's an example where we have mice housed in 2s. And in one cage, we have 2 mice that are control mice, where nothing will happen to them. They'll just socially interact during the 1 hour. And in the other group, we have 1 mouse that we assign as a pain mouse, and the other will be the bystander. So we assign these groups, we match them based on their Von Frey thresholds at baseline. And then 24 hours later, we inject CFA into the CFA mouse, place them in all of the cages. So you have empty, clean home cages just sitting on the counter. You can videotape this if you need to. You can modify this with like a sort of tall open top if you have optogenetics or something like that, where you have tethers coming out. And you just allow the pain mouse to interact with their previous cage mate for 1 hour. In the control condition, nobody gets any injections, no control injections or anything like that. It's just 2 mice that know each other interacting for 1 hour. Immediately after that 1 hour social interaction, the mice will then be subjected to whatever types of tests that you're interested in. And so in this case, I'll show you mechanical sensitivity, though we've done all types of tests, which I'll mention in a moment. Okay, so so for the CFA, we have given multiple types of inflammatory stimuli into the paw. CFA is a long-lasting chronic arthritis-like pain, and you inject this into the interplantar surface of the hind paw. But you can also use formalin, capsaicin, carrageenan, any of these inflammatory mediums to induce the pain. And with that, so successful tests that besides mechanical sensitivity also include thermal and chemical nociceptive sensitivity, so something like formalin or a hot plate. We've also done anxiety tests like elevated plasmase. We've looked at affective behavior with emotional discrimination, anhedonia, behavioral despair, et cetera. You name it, you can pretty much run any behavioral test after this paradigm. Okay, and so speaking a bit about the housing again, so it is critical that you have these mice housed either 2 or 4 per cage. And so this is just an example of the pairing during the social transfer. And then after the social transfer, you need to split them and house them in their treatment matched groups. So you can see up here on the top, the control condition has 4 mice, and you split them up during the social transfer into 2 groups. Mouse 1 and mouse 2 interact. Mouse 3 and mouse 4 interact. And then afterwards, just to match the conditions that I'll show you in a moment, you're going to house mouse 1 and mouse 3 together, mouse 2 and mouse 4 together in brand new separate home cages. And this is why, because in the pain condition, you have your 4 mice in a group. 2 of these mice are going to get injected with CFA, 2 are going to act as the bystanders. But after the test, you cannot house them back together again because there will be a continual social transfer of pain. So you're then going to house the CFA mice together and the bystander mice together. And so you're really trying to match that with the control conditions by switching, their partners from the social transfer into their other cage mate partners. Okay, and so this is just a bit of representative data from the STAR protocols paper of what this looks like. So again, with mechanical threshold, any decrease in their threshold is indicative of hypersensitivity or pain. And I'll say now, probably a dozen researchers have replicated this phenotype across 3 or 4 different universities. We've had undergrads, grad students, postdocs, all replicate this phenotype. And so what you can see is Control mice really don't deviate from their baseline, which is represented by the dotted line, if you measure this across time. So immediately after the social transfer is this time point 0, and you can measure a time course 4 hours, 24 hours, 48 hours later for as long as you like. You can see that CFA mice show a robust hypersensitivity just 1 hour after that CFA injection and social transfer session that lasts for basically as long as you can measure it. And so depending on the type of pain induction here, you may or may not see chronic pain, you may see an acute pain state. And the most important part is that these bystanders really do show a marked hypersensitivity that's almost as robust as the CFA mice when you measure immediately after that social interaction. And in our hands, this seems to last for definitely 4 to 6 hours and begins to go back up to baseline levels around 24 to 48 hours later. And so even though the induction of this pain via the social interaction was only an hour, the pain state lasts for almost 24 hours later. And this is just an example of that data at time point 0. So you can see the CFA and bystander mice really look very similar. Even though the bystander mice did not receive any pain injection at all. So these mice are really displaying, a pain state that is more indicative of the CFA mice. It looks like they got injected with a painful stimulus, even though they did not. All right, and that leads me to the social transfer of analgesia, which is sort of a modification of the social transfer of pain. So we really wanted to look at something that wasn't negatively valence. We wanted to look at empathy processing and something that was positively valence. So we looked at pain relief, which is a rewarding experience to have relief from pain. And the paradigm is remarkably similar with a few key differences. And so again, you have your control mice, and then you have your analgesia mouse and your bystander. But the difference here between the paradigms is that all the mice in every group receive CFA. And this is because the thing that we're interested in is the pain relief, okay? So both mice in the control group receive CFA. They're both actively in pain. Both mice in the bystander group receive CFA. The only difference is that one mouse, our target mouse, receives an injection of morphine. And then the mouse that receives CFA interacts with that mouse that got morphine. And morphine is analgesic and relieves that CFA pain. Then you do your mechanical sensitivity. Unfortunately, you cannot do mechanical sensitivity testing on morphine mice because they're super hyperlocomotive. And so we used thermal nociceptive testing. And we also found that the CFA mice don't develop thermal hypersensitivity after 1 hour. It takes a little bit more time. So in order to look at the thermal hypersensitivity and be able to include the morphine mice in the nociceptive assay, 1 week later, we subjected them to another social transfer session where we just poke the mice on their feet again to elicit that active pain state, inject morphine 1 more time, and then place everybody on the thermal place test. So in these ways, you have to pay attention to what type of nociceptive behavioral test you're doing and the time course of that pain stimulation. But what we see is that here you can see in the mechanical threshold, again, we don't have that morphine group represented, which would be up here somewhere. But you can see that the CFA control mice are very hypersensitive. The CFA bystander mice are still hypersensitive, but they're not nearly as hypersensitive as the CFA controls. They're requiring some pain relief from their morphine partner. And this is just that percent time on the warm floor here in C, where I show 1 hour after CFA. The CFA mice don't show thermal hypersensitivity, but 1 week later they do. So if you look at percent time on the warm floor, so they tend to avoid the warm floor if they're in pain, represented by the CFA controls here. Morphine reverses that produces analgesia and reverses that pain phenotype. And the bystander mice look remarkably like the morphine mice. They do not look like their CFA control counterparts. So this pain relief is acquired by that 1 hour interaction with a mouse that received morphine analgesia. Okay, so that's the social transfer of pain and analgesia. And we believe you can use this to measure empathy, emotional contagion, and the social communication of pain. We've done quite a bit of optogenetic experiments and neuroanatomical tracing experiments to show that the brain regions activated in the bystanders is remarkably similar to humans experiencing empathy. And we've now used this in mice, rats, and even prairie voles and showed that it's valid across all these different species. And this paradigm is very easily adapted for use with optogenetics, chemogenetics, and even calcium imaging and mini scopes. So with that, I'd like to thank STAR Protocols and Open Box Science for hosting this today and for publishing this protocol paper. I hope that people use the protocol. Definitely reach out if you have any questions. I'd like to thank Ben Rain and Aaron Jones, who are the first authors of the STAR Protocols paper, and the others who contributed to some of the data in my lab, as well as Rob Malenka and Nawaz Sada, who were authors on the original science paper. And then I'd like to thank our funding mechanisms for this, and then quickly mention that the Smith Lab is hiring at all levels at UCSD if anyone is interested in working with us on this. So thank you so much.
Yeah, thank you so much, Dr. Smith, for this fantastic talk. It's really very impressive. So now I guess we can move on to our Q&A sections. So is there anyone from our audience having any questions? Oh, yes, Vivian, please.
I saw Jerry's hands up first. Jerry, do you want to go first?
Okay,
No,
Jerry
you go
first.
first.
Yeah.
You go first.
I guess I have two questions. The first question is, I was really impressed by this analgesia effect, and I was wondering the length this analgesia, does that depend on the social transferring time? Like how long you have interact with the animal, does that count, or what would be your suggestion if you wanted to test how to elongate or how to sort of strengthen this social transferring effect? What would be the most critical step to tweak? And my sorry, yeah, let's go with the first question.
Yeah, let me answer that one because otherwise I'll forget. Okay, so we really fully looked at a lot of ways which of the time course in the social transfer of pain. We tried 30 minutes, we tried 2 hours, we tried 4 hours, and it didn't seem to make a difference. 30 minutes seems to be enough. I will say 10 minutes is not enough to acquire the social transfer of pain. We didn't go into that level of detail with the social transfer of analgesia because we developed it second. So we just looked at the 1 hour. We saw that that was sufficient, and it lasts for 4 to 24 hours later. So it lasts quite a while, that this analgesia that they show, they show the pain relief for more than 4 or 6 hours after that. But in terms of like, how short can you make the time period? I actually don't know the answer to that. So we might have to characterize that more fully. Okay.
What was the
Yes.
second question?
So, yeah, you have a second
Sorry.
question.
Yeah. Like I was just wondering, have you record the interaction during the social transferring time and then see what kind of interaction actually are critical for this? Or I guess a related question would be, if you remove any sensory input, like it's in a blind mice or in a mice that cannot hear or smell, would that actually find the most critical sort of sensory input to help with this transferring?
Yeah, fantastic question. We get that all the time. So in some of so there's a sort of 3-part answer. In the original alcohol withdrawal studies, we showed that the bedding, so just the olfactory cues, were sufficient to induce hypersensitivity. We've now shown that again, that 1 hour exposure to dirty bedding from pain mice is enough to induce the hypersensitivity. But the olfactory cues, I will say, I haven't replicated this yet, but it seems like it induces the pain hypersensitivity, but maybe not some of the affective and emotional changes, which don't quote me on that until we replicate it. But it seems like maybe multi-sensory input is important. We have used zinc to ablate the olfactory processing in the bystanders, and this does prevent the social transfer of the mechanical sensitivity. And we're now we're going to try blocking visual cues. And then to your last, and so we don't know the answer about visual yet, but to your last point, we have recorded the social interaction. We have tried scoring those videos, but they're hour-long videos. It's super time intensive to score those by hand, and nothing has jumped out at us. There isn't a significant amount of grooming that we can really see. The interaction time and the way that they interact doesn't appear to be changed, but we are going to start using some automated behavioral tracking to see if we can get some more nuanced behaviors that might be caught by like actual computer tracking. So yeah, thank you.
Thank you. That was very interesting experiment design. I have the same question as Vivian, but I have one additional question. Is this
Sure.
sex and age of the mice affect the degree of social transfer of pain?
Yeah, so fantastic question. This happens in both male and female dyads. There doesn't seem to be a difference in the magnitude of effect, although I'm not convinced that would be true in every scenario. And what I mean by that is CFA is a really robust pain stimulus, and the females acquire it just fine. But I think the bystanders are kind of like as low, they're almost at a floor effect as they're going to get. And if you used a less intense pain stimulus, you might see a sex dependent effect that you could pull out of that. The one caveat is that females are more hypersensitive at baseline and they show more robust pain responses overall. And so there are sex differences in the pain phenotype itself, but not the magnitude of the social transfer. Again, we haven't thoroughly tested, whether or not females might acquire this faster, whether or not they might show sort of more robust other types of behaviors. And those are things we're actively exploring, because for this to be a really valid model of empathy, there should be some sex differences, right? It's known that in humans that females tend to show more empathy behaviors as well as in foals. So it's something we're actively exploring, but we haven't seen any sex differences. In terms of age differences, I've done this in mice, basically young adults, like 6 weeks, all the way up to because some of our recordings were in 20-week-old mice, and it works the same, though I've never, sufficiently powered those experiments to look at age differences. And I am very interested in looking at adolescent mice to see, because adolescent mice are more social, especially males. And I think we'll probably use the adolescents to explore familiarity versus non-familiarity. We were kind of worried about having unfamiliar males together because of the fighting. So we never have looked at that. And I think using younger mice might be a way to really look at those things.
Thank you very much.
Yeah, no problem. Any other questions?
I think there's one question in the chat.
Oh, okay. Yeah, what do you
yeah,
think about mirror neurons and the relationship with social transfer of pain? That's a fantastic question. It's something we're actively exploring. We're going to be using miniature microscopy to see if the neurons that are activated in the anterior cingulate cortex during self-pain and observed pain are the same. So I hope that that's the answer, but I don't have an answer to that yet. So we're actively pursuing that. And then one more question. Is it possible to differentiate the social pain transfer from stress or alertness more than pain? So in the original alcohol withdrawal experiments, we thoroughly characterize the stress responses. And I'll say we've done a bit of that with the rapid social transfer. There is a very transient potential increase in corticosterone, but it disappears pretty quickly. By 24 hours, there's no more increased corticosterone, although the pain persists. There's no effect on elevated plasmase or anxiety tasks that we can see. It's not like a hypersensitivity of all sensory systems. So they don't show changes in acoustic startle or other sensory responses. So it does look to be relatively pain specific. There are changes in things like behavioral despair and anhedonia, which isn't published yet. And so we think this is a multi-dimensional pain experience, though it doesn't seem to be dependent on stress systems per se, or at least persistent activation of stress systems. And so we're interested in stress, but it doesn't seem to be like a hypersensitivity of all sensory systems, or it does. And one more thing. If you give metirapone, which is a corticosterone blocker, or something like diazepam, which is an anti-anxiety drug, this does not block the social transfer of pain. So it really doesn't seem to be dependent on stress systems. Okay.
Yeah, thank you very much. I think Mingyu is back. Maybe she has some connection problems.
Yeah, so sorry about that. Yeah, I have some connection problems. So earlier I saw there's a question from chat. So is that?
Dr. Smith answered both of the questions.
Okay,
yes.
great, awesome. Yeah, that's a really fantastic talk. Yeah. I actually also have one question. I don't know if it's okay to ask. Yeah, I'm just curious about this transfer, for example, other types of emotional transfer like stress, can that be applied to this kind of test. And also like drug abuse, like some people, they take drug and have some kind of, emotional change. Can that also be applied using this protocol?
Oh, fantastic question. So yes, stress, definitely. So some people from the Hong lab at UCLA have shown really beautifully recently that if you shock a mouse and then pair it with a partner, the partner mouse will just display consolation and grooming behavior. And there are lots of other examples of this like social transfer of stress, or even social transfer of analgesia related to stress. So social transfer of distress in itself, primarily using shock paradigms, is pretty well characterized. So that is something that's definitely possible to look at. And I think that the mechanisms might be slightly different from the social transfer of pain. And then the drug abuse thing is really fascinating because I tried, and now Asada, for about a year or a year and a half to get the social transfer of reward, or a social transfer of condition reward, or social transfer of euphoria after drug taking or drug seeking. And it wasn't something that we have been able to robustly show, but it's something that I will continue to pursue because I think we may be able to figure out how to get that to work. But if anyone has any ideas, I'd love to hear about that. Okay.
Okay,
Maybe I still there?
fantastic.
Okay.
Yeah, thank you so much again for this really awesome talk. Very impressive. And
Yes,
I will
thank you for having me.
Yeah, and I would also like to thank all the audience for joining us today, and also the Open Box Science for this hosting this seminar with us. So I guess we look forward to seeing all the audience in the future Open Box Science seminars. And also one more thing, like for STAR protocols, we are actively looking for reviewers, volunteer reviewers. So if you are interested to serving as a reviewer for us, welcome to register. I have already posted the link on the chat. So we are looking for any bench scientists, including postdoc or PhD students. So anyone welcome if you like to have review experience. Okay, so I guess we can end the seminar for today's talk. And again, thank you for the speaker, Dr. Monique Smith. And thank you, Jerry and Vivian, so much.
Thank you. Bye-bye. Have a good day.
Thank you. Bye.